Hello everyone, we're just waiting for a few more people to join. If you haven't, please already have a look at the pre-tutorial instructions, and I'm going to paste the link in the chat in a second as well. Okay, hello everyone. It's 1 o'clock now. I don't know if other people are going to join us or not, but at the moment it looks like we're a very nice kind of small crowd. So if you want to share your video, please feel free to do so. It would be nice if we kind of have a little bit of interaction today. We're already losing people when we say that. So hi everyone. I think we'll just make a start. So I'm Elisabeth. I'm the main host for today, and I've got 3 co-hosts with me today, who are Eva-Maria, Karim, and Sara. And so I just put a link into the chat for a tutorial kind of pre-guide. I don't know if you had a look at that, but if you can have a look at that, that would be great. And while some more people are joining as well, if we can download the data, that would be great as well. I'm just seeing. Yeah, fantastic. Thank you, Nikki. All working great. So I'll just put the link for the data into the chat as well, because I don't know if everyone has downloaded it. So if you could do that, that would be great. Let's quickly walk through the Fiji, how we get it ready as well, if that's okay. So I just need to share my screen as well. So in Fiji, we need some update sites, and I don't know if you have done that or not, but basically if you go to update, we need to update 3 different update sites, and that's again in this tutorial guide, which is somewhere here, opened this one. So they are specified here, and you can do this with Fiji update. It's running a lot of stuff, I'm sorry. Might not run that much stuff for yours. Okay, and then if it has done all the other kind of local updates and stuff, you can go to manage update sites, and we need 3 of them. The first one is this 3D ImageJ suite. The second one is the neuroanatomy, which is far down here, and then the third one is this IJPB plugins, and you just tick that, you close, and then you say apply changes. Once it has done all the updates, you just need to restart Fiji, and then we've got this so far ready. So that's what we need in terms of the Fiji side. The second thing was the code that I've linked you to, the data that I've linked you to, and the third thing that we're going to need is actually the code. So I'm going to put that link also in the chat for you. And sorry, without the hashtag, obviously. And if you go on this side, so this is my GitHub page where we collect all the code. We have a little bit of an introduction, and it has also all the links to the things. So if you kind of want to read a little bit more about it, all of this will be in here. And for the code, you just need to download it as a zip. Obviously, then it downloads it, and then you can extract it wherever it downloaded it to. And what you will have at the end is a folder that's called I2K, and we'll go through these dates today. But please, if you want to follow along, just download it now so we can follow everything step by step, if that's okay. And can I just get quick of like yes or no in the chat if all of that so far makes sense? That would be great, so that we have a little bit of feedback of what's going on. So far things clear? Yes? No? Maybe? Okay. Okay. I think so. If anything is unclear today, just put it in the chat, raise a hand, interrupt us. All of this is meant to be really interactive and very informal, and we want to make sure that people know kind of what we're working through and what it's all about. So before we kind of go into the individual sessions or like the individual sections, I don't know if Karim, Sara, and Eva-Maria just want to briefly say hi, who are we, and then we can kind of start with the actual session, if that's okay. Yeah? Sara, do you want to start?
Hi everyone, my name is Sara. I'm a medical student at UCL and I'm also part of the MacDonald labs, and I've been working with Ellie for a while on the GliaMorph tool. It's very nice to be here.
Karim?
Hi there, my name's Karim. I'm also a fifth year medical student, also with MacDonald Lab, and been working with Ellie on this GliaMorph toolkit. So I really hope you enjoy today. It's been really fun working over the past few months. Hope you enjoy.
Hi, my name is Eva-Maria. I'm a master's student and neuroscience master's student, also MacDonald Lab, and I'm working on the regeneration in the killifish. So regeneration of the retina in the killifish.
Fantastic, thanks. If all of you, because I obviously don't know you, if you want to just put in chat if you're like biologists or developers, if you're advanced Fiji users or very kind of early in Fiji use, that would be great. So we kind of get a little bit of an idea of who you are and what you're interested in. And while you're doing that, I'll start with introducing the model and what we're working on, if that's okay, and kind of just give you a little bit of an overview about what we're going to work through today. So just put in chat, you know, who you are, what's kind of your interest. That would be fantastic. So, right, now that we've talked through kind of like what we need for the download, that's fantastic. And so today we're going to talk about GliaMorph. So it's a toolkit that will allow you to quantify Glia morphology in 3D. And so today we have kind of a little bit of introduction about the model, so what we're going to work on, and then we will have a section about data understanding and image comparability, which Sara will lead. And these are really for you to understand your data in 3 dimensions and what we can do to get better data quality, but also why it is so important to have really good data quality for your actual quantifications. We then talk a little bit about the image segmentation, and I appreciate that this will be very different to different biological questions, so we can have a discussion at the end how we can improve this and adapt this to your data. And then we will talk about how we can actually quantify different features in our data. And lastly, we'll talk about data interpretation, and again, how this might inform your data workflow from the very start. So what we're working on are retinal glia cells called Müller glia cells, and these are a form of support cells in the retina which have a very specific shape, as you can see here, that allow them to interact with different cells in the retina, such as the photoreceptors, the neurons, or for example blood vessels. And so their shape allows us really to understand how they interact with other cells, but also how, for example, they are in terms of maturity, but also in disease. So for example, in disease, you would see an alteration of how the cell nucleus looks like, but also how their interactions with certain cells will be changed. And so we use their shape as an outreach for their functionality. And so this is really where we need this morphology analysis to say what are they doing and how healthy are they, etc. As a model, we are using zebrafish, and we mainly use transgenic lines. So this means that we have genetically changed the fish so they express certain fluorescent proteins that allow us to visualize these different cells of interest. For those of you who are not working with fish, we can do the similar kind of experiments with mouse and human data, but in mouse and human data, we would obviously more use antibody stainings rather than transgenic lines. And so for today, we're not going to talk about the biological kind of nitty-gritties, but just kind of, if you have questions, obviously we can talk about them. But today is really more about the shape and the shape analysis. And so kind of we have an apical part and a basal part, and within these structures we have 5 different subdomains, whereas 1 and 2 allow them to interact with photoreceptors. Number 3 is where the DNA sits and contains the cell body. Number 4 is probably the most interesting or mostly studied region, because this is a region where they interact with neurons, which are basically here in a layer called the inner plexiform layer. And so this is a highly studied region because there's obviously this interaction with neurons. And lastly, they have a specialized structure called the end feed. And so understanding what each kind of position does is obviously hugely interesting to the biologists, but for today, I just want you to kind of keep in mind that each one of these subregions has really an important function, and obviously each of their shape is very important to be understood. And so what we do to study these data is we use AiryScan microscopy. So AiryScan is a form of microscopy where we have 32 elements of the detector working together, so we can push beyond the diffraction limit. And that's hugely important because obviously these cell features are very small, especially these subregions that interact with these neurons in this layer called inner plexiform layer. And so AiryScan is basically a semi-super resolution microscopy technique that allows you to resolve a lot of these fine details. And this is what we use for the data today. But also we need to keep in mind that obviously because it is kind of in this family of confocal microscopy types, that because the laser goes into the tissue, you have a very quick kind of loss of data within this stack. And so we will talk later a little bit about this, what this means in terms of processing, but it's just for now something to keep in mind. And so for today, we talk about this GliaMorph workflow. We've just put it on bioRxiv a few days ago. I'll put the link also in the chat in a few seconds. But basically what we'll do is we'll take some raw data and we'll go through the whole pipeline today using different tools and understanding how we can go from raw data to something that's meaningful in quantification, such as, for example, how large are, or like how high are these glia cells, what's their volume, what's their kind of branching points and things like that. So something that's meaningful for us as biologists to really interpret the data that we're looking at. And each one of these different subsections will be discussed by someone in the team today. And with this, I just want to check in if there are any questions, and I'll just have a sneaky peek at the chat, what everyone is saying so far. So if anyone has a question, just unmute yourself and just ask ahead. I love that Laura says she's 1/4 biology and 3/4 developer. Yes, I think that's pretty much the same for me. Fantastic, thank you. So if there are no questions so far, I think we can start with Sara talking a little bit about data understanding, if that's all right.
Let me just share screen. Just one moment, there's a privacy setting that I must change.
I've changed. Can you try again?
I think it's in my case, it's on my side that I need to change it, but it should be fine. Okay, I might need to restart. If you give me 1 second.
Yeah, no problem. All good. So hello everyone who has just joined in and who I haven't seen so far. It's really nice to have you all here. Again, if you feel comfortable, you can just show your video. It's nice if we have a little bit of feedback. If not, obviously leave it off, but it's nice if we have a little bit of interaction.
All right, I'm back. Apologies for the delay, and let's see if this will work. I think it will work just fine now. All right, good. Can you all see and hear me?
Yes.
Lovely. Okay, so let's start. So whenever you start your work on a new data set, it's always important to gain an understanding of the data that you're working with and whether they match your expectations and your requirements for further processing. So what we see here is an animation, so it's labeled as stack. So this refers to the 3-dimensional images. So we're working with 3D images collected by AiryScan, and they are a stack of 2D slices layered on top of one another. The animation that you see moving here shows you the different 2D slices as you go down the depth of the stack. Okay. The maximum intensity projection that we see here is an algorithm that will select pixels of the highest intensity from every slice throughout the volume to construct an overlapped 2D image. And this here just shows you the stable transgenic line that we're working on. So this is the TP1 venous pest, which visualizes notch signaling in Müller glia cells in the retina. So each of these cells is composed of a very intricate structure, which Ellie will explain. And just to orient you guys, this is the apical region of the cells, and these are the basal end feet. So it's a bit like upside down. All right, so now we know kind of what we're looking at, but what should we look for if something is going wrong? What should we inspect for? So a 3D image consists of 3 dimensions, X, Y, and Z, Z referring to the depth. So whilst in 2D images, we refer to X and Y dimensions and a pixel being the smallest unit, in 3D images, we will be referring to voxels. Due to resolution limits, the Z dimension is greater than the X and the Y. So as we can see here, we obtain this type of image, so more rectangular looking shape rather than a perfect cuboid shape. However, sometimes when you do save your data, it can automatically be stored into this isotropic version where all of the dimensions are equal. So we need to make sure that whenever our data is saved, it is saved in this format with Z being the largest dimension rather than this. And we can do that by inspecting the 3D rendering of the image using one of the plugins that we've already installed, the 3D viewer. So if you compare here on top, you have the anisotropic voxel. So when the 3 dimensions are not the same and the Z is the largest. So this is what it looks like. And when you compare it to a false 3D rendering where all dimensions are 1, 1, 1, if you see it is a bit squashed, so it's not proportional, it's not looking correct. And if you do then later proceed to perform more analysis on this lower version here, the data that you'll collect from it will not be right. Okay, so now let's see how we can do this in practice. All right, I'll ignore this for now. We've got this data set here, the folder that we've all downloaded from Zenodo. We can see here there's 3 different TIFF images. These are the ones that we'll be working on. And then this folder here contains the output. So at the end, after we've run all of the steps on our 3D images, we're meant to have something like this. So the tutorial prep is not what we'll be working on. This is what. Okay, so what we do to work with Fiji is you can take an image, just drag and drop it here. Okay, and you can see a similar stack to the one that we saw on the presentation. Apical domains here, basal and feet are here. Okay, and if you scroll down using the slicer, using the slider, you will go further down the slices along the Z dimension of depth. All right, so an important point to check for here is Z-axis decay. So due to point source illumination, when we're taking these images using the microscope, we have a problem with tissue penetration. So sometimes the top layer can be of very good quality, very strong signal, as you can see here, but then the deeper layers might have poorer signal due to lower penetration. So what we do by just checking with the slider is we can investigate for that as well. So it's an important point. Okay, and now to get a maximum intensity projection, what we can do is go on image, Z stacks, so image stacks, Z project, and you just click on OK. And this is what you see here, an example of what we saw on the presentation. All right, and now let's visualize, we can close this. Now let's visualize the image. And now actually, before that, we need to check the image properties. So as we said, we need to look for the X, Y, and Z dimensions. So we go on, image properties. And this is what we're looking at. We see different properties of the image. Channels simply refers to the color channels. In this case, we've only used green, so there is 1. Slices is 64. This refers to the 64 two-dimensional slices that are stacked together to form the 3D image. And frames refers to the time frame, so the point in time where the image was taken. This is not a continuous image throughout time, so this is taken at one point, so that's why frame is 1. Then the X, Y, and Z dimensions are here pixel width, pixel height, and voxel depth. And we can see here that they are correctly saved in the right proportionality instead of being 1, 1, 1, which would be wrong. Okay, so now to inspect this in 3D, what we need to do is go on plugins and we can find the 3D viewer there. However, because my laptop would not be able to process this large version of the image, I will simply get a cropped version that we will show you how to get when you use the later steps of the GliaMorph tool. So we'll use this. So, okay, this is a cropped version of that same image. And to investigate, to inspect this in 3D, again, you go on plugins, 3D viewer. You need to give it a little bit of time. It might be a bit slow. Okay, okay. Oh, she's giving me an error. Let's try again. Okay, we need to be patient. My laptop is running Zoom and Fiji at the same time. Okay, let's try again.
If you want me to share for you, just let me know.
Yeah, if you could simply slide one of the images, I can do stop share for now, just while my laptop kind of recovers. Okay, yeah, so you can just slide one of the cropped images onto, okay, yeah, so it's gone a few steps ahead. Yeah, so now we were just waiting for the 3D image to appear, and this is what you get. You can use your mouse to move the image and look at it in different angles. If you double click on the mouse, you'll be able to get a bounding box that just highlights the edges of the 3D image. All right, so this is what it should look like. And now what would we see if it was a incorrect version, so with the 111 dimensions? We go on image properties, you change dimensions to 111. Okay, click OK, and we do the same thing. So we go on the 3D viewer. All right, and you will get a 3D version of the image that is not going to look right. It will look a bit squashed. So if you put the bounding box again, you can very clearly see that there is this proportionality there. Okay. All right. So this is the first part about data understanding. We can move on to the next slide. Thank you, Ellie. So this bit is about image comparability. So now that we have an understanding of our image and we can see what it should be saved like correctly with the correct proportions, we can now proceed with the next step of analysis. So whole mount zebrafish imaging, just like many other biomedical models, requires sample embedding. However, there are no hard set lab standards on sample orientation. So as you can see here, there's 3 different alignments for the first 3 images. They're aligned in different directions. They're not all the same. And this variability in position can affect our data. Similarly, fields of view that are required can be larger than needed, and images cannot be directly compared to one another. As you can see, the middle image has a greater depth than the other 2. So also the Z depth, the Z dimension is not standardized across all of our samples. So the subregion tool allows us to kind of standardize all of these different samples and go to what we see here on the other side. So if we click on the next. Yeah, exactly. So what we do with the subregion tool is extract a region of interest by using a line selector, which you see there. It will then rotate all of the images using that line as its guiding orientation. And it will create a bounding box around that line to highlight a region of interest, which we call ROIs. And it will then crop the X, Y, and Z dimensions around that region of interest. It will do this in a standardized way for all of your images. Okay, so we can show the group how to do it now using Fiji. Ellie, would I possibly still use your screen and just comment as we go?
Yes.
Okay, so you open the folder, you can drag one of the images onto Fiji. Click, okay, let's just wait for a second. All right, and when it opens, you click on the line selector, which is there, and you draw a line from the inner part of the retina extending outwards in each image. It's important that you select a region with the best signal data because this will become the center of your region of interest, yeah? So then you go on Edit to save this region of interest, Selection, and Add to Manager, which you see below there, exactly. All right, it has already been added, but if you don't see it there, just click on Add T on the side. All right, we will repeat this for the next two images. An important point is to make sure that you do it at the same, you repeat this for the images at the same order that the images are aligned on the folder, because you want the regions of interest to match the order of images. So we know which region matches which image. Okay, and always when you drag the line selector, always do it from one side to the next. So if you're starting from inwards out for the next image, don't do it from outwards in. Always keep it standardized. Okay, all right. So once you've saved all of the three regions of interest, we go on More at the side and click on Save, exactly. And we will save this as ROI set line. It will be saved as a zip folder on your original folder. You can see it there. There it is. Perfect. Okay. So now we can process the images using the subregion tool macro. We go on macros, find the subregion tool. We drag it on Fiji. Yeah. And you click on Run. And what you'll see there are some output dimensions. It is missing output image height. This is because the image height is what you have selected with the ROI. That is what this will measure. The width is a default of 60 micrometers. It's meant to include 12 microglia cells at the age of 5 days post-fertilization. So it's giving a standardized amount of cells within each ROI. The depth is also by default 10. And the sigma size is a parameter that accounts for the retinal curvature. And this is also set at the default of 10 micrometers. So we can click on OK. And we select the folder where we want this to run. And this will be the I2K folder, exactly. Whenever you run a macro on a folder, it will run on all of the images inside this folder. So you don't have to worry about selecting each of the individual images one by one. And also, I think there was also the other folder, tutorial prep, within that large I2K folder. It will not run on that. It will only run on the images. So you just have to wait until the macro is done running. And at that point, you will get a notification. Yeah, macro is finished, exactly. All right, so what you can see now, if you go back to your folder, you'll see some new files there. So the first one is MG height. These are the retinal heights that you have measured using your ROI line selector for each image. Additionally, you will see the folders XY deer and Z deer. So for Z deer is the one that we'll be using. This means that all the images here are rotated along the XY axis and reduced in the Z axis direction. And if you open them and drag just one of them on the Fiji or a couple, you can see how they're all now uniformly oriented, and comparable in size. So this is the subregion tool. I think Ellie now can take you through the next few steps. Thank you, guys.
Okay, thank you so much, Sarah. Sorry for all the technical glitches, but we'll manage today, kind of. Can I just check in if people have any questions so far or any comments on that? Either raise your hand, unmute yourself, say something, or put it in your chat. It would be nice to get some feedback at this point.
I guess I'm sideways. Can I ask a question? So, okay, I'm at your GitHub and you have all the different macros. And so are you actually running the macro steps from the macro folder, or is it actual plugin? I'm a little bit confused about the steps on actually how to run all the different steps.
Okay, thanks for that great question. I think, yeah, we should have clarified that a little bit more at the start. So when we download the code, so what I've done is everything is an individual macro at this point because it gives you more flexibility to apply individual steps. So it's not, you know, hard kind of for one certain application, but that it's more flexible. And so for today, we're just talking about these macros in the macro folder, and each single step will basically be covering, sorry, I just need to reopen this. We'll be covering one of these steps. So for example, we now have gone through this subregion tool and we took the original data and just made them the same size. That was kind of the first step. For today, we just have a single color image, so we're not going to talk about the split channels tool. But the next thing that we're going to talk about is actually how we can look at the data in a little bit more detail with the zonation tool, which is basically plotting these graphs that we're looking at in a moment. Does that answer your question?
Not quite. Actually, it's more of a technical question of,
right,
so
sorry.
I have to open each different macro for each different step I want to perform and run that macro specifically?
Yes.
Okay, that answers my question.
Yes, so because the thing is, I find it easier if people just do one step at a time and then look at their data and see, you know, has it worked, has it not worked, are there any problems, and then work to the next step. Because when I had like multiple steps in one go, people were just confused, tried too many things at once, and things.
Okay. I understand. It's just in my lab when I'm dealing with general biologists, and I give them something where they have to deal with macro script at all, they freak out. And so I'm trying to I'm just now starting to work with imaging macros and building and developing them for my own analysis purposes. And so, and I know I built a simple one where, you know, all your variables are at the top, just put your variables in at the top and push run. And they're like, oh no, I can't do that. So I'm just trying to understand the level of complexity here. So
yeah, no, thank you. So that's also why we have basically you drag and drop it, you hit run, and if anything is to change, it will be just in a graphical user interface. So we don't want users to actually having to change anything in the code, because all of us are kind of from a biology medicine background as well. So we absolutely understand this apprehension of like, oh my God, I can't touch the code kind of thing. But still, we wanted to keep it open enough that if people want to directly change something, then they don't have to be able to work with a plugin code, but actually can work with a macro code. So I think for us, that was this trade-off of like biology user friendliness, but still adaptability, if that makes sense.
And so my last question, she was the last step where we're talking about the voxel depth size and the sigma orientation because it's the retina and the curvature. So can that be changed? I mean, it can be changed, but how do you know how to change it when I'm not dealing with the retina, I'm dealing with the cerebellum and I'm dealing with single, you know, glia versus something that's actually in a tissue of the, you know, connected tissue like that?
I think on the one hand, it's test, trial and error, because obviously each data set is different. It's already with different ages, the retina will have different curvatures. But we found, so in these ages that we look at, these first 5 days of development, we find these 10 micrometers to be actually enough, so to speak. But already at like older ages, when we actually have less curvature, we can make 5 micron, for example, instead. But that's a lot of like knowing your data as a biologist. So for example, in the cerebellum, you might not even want to use this, but might use a different approach to actually extract the region of interest. Okay. But that's again, like I think trial and testing and seeing, you know, when does it make sense and when doesn't it make sense for your data.
Okay. Thank you.
Thank you. I also have some comment from Frederica. Just reading that. So maybe some input about the macro, because I noticed this while running the code right now. It might be helpful to close all open images and clear the reach of interest. Yes, thank you very much. Yes, so before we always run a new macro, we normally just close everything in the background. It's similar with like, once you hit run, don't start clicking about and, you know, faffing about with Fiji, because I think that's also something that we tend to do with like, oh, we're impatient and start clicking about. I would say when you start something, just grab a cup of coffee or, you know, start writing on a document and don't click about in Fiji. That's generally a bad idea, because Fiji works the way that if you click on it, that's basically selected and it would overwrite what we have selected in the macro. So normally you get an error, and that's kind of a standard thing to do. So yes, clear everything, start running your macro, and then just leave it be until you get this message, macro ended or has finished. Okay. Any other questions, or are we all good? Okay. I'm just checking where we're at this moment. Okay, so what we're going to do now is actually, so one thing that we have implemented in GliaMorph is the ability to take 3D data and convert them into 1D vector to look at them. And this is what it looks like. So in our case, the Glia cells have a very nice apical to basal polarity, and it has these 5 different subdomains. And so what we said is, from a biology point of view, if we have stainings like these or transgenics like these, we have obviously a difference in intensity distribution. And so we could make use of this to take a 3D stack and actually describe the images as a 1D vector. And so all that we do in Fiji is we automatically take these stacks, so we have them normalized now, and actually compress them so that they are 1D vector. And we're just going to do this now because I think it makes more sense when you actually see it. So what we do is basically take the next, or the step 6 tool. So in this case, like I said, we're going to jump over the split channels tool because we only have one marker in this case. But if you have multiple markers, you can split your channels beforehand. But now what we're going to do is we drag and drop this step 6 sonation tool. And in this case, we again just hit run. And it will ask you a few questions. So first of all, do you want to do this? Yes, we want to do this. It will ask you about scale normalization. And this is something that, again, depends on your data. So what this means is that if you look at, for example, different age groups, it could make all of them the same height. So for example, if you would have naturally smaller retinas, it could stretch them automatically to all be the same size. This makes sense if you want to look at very local differences, but if you're actually interested into how this distribution changes from the top to the bottom with respect to the whole scale, then don't do the normalization. But generally we say, if you have new data, run it with and without, and then, you know, have a look, what does it look like in your data, what does make sense. But in this case, we just say no for now, and we just leave all of this. So in this case, this would be to what you could stretch your data if you would do the normalization. And this sigma is what we've added before. And so this is just so we know, you know, have you added anything in this step before or not? So in this case, we just leave it because we haven't touched it before. And so we just say, okay. And in this case, again, we select the input data. And in this case, what we do now is use this ZDIR. So this is the output directory from what we had from this subregion tool. So this means that our data are all the same size, and this means that we can produce these 1D vectors. So we can select, and then in the log, it will tell you what it's doing in the background. And then again, just don't touch Fiji, just let it run. And now it's just going to do this 3D compression into a 1D vector, and then again it says macro is finished, and we can go to our folder. So in this case, we have now used this as an input, and so the output directory is this sonation tool. It will also tell you that this is the output directory in your log file. And so if you would look in here, what you have is, it just tells you kind of what data it has used, and it will have produced these 1D vectors just as visual representation. And zonation tool result, in this case, it just again tells you the image height as well as the basically length of this region of interest. So image height is this region of interest that we had before plus this 10 micron sigma, and length of the region of interest is basically the image height without these 10 microns. But the thing that we're interested in is the zonation tool profiles, and in your case you can open that with anything. It's just a CSV file. But what it will have done is basically plotted all your data from the top to the bottom as intensity profiles. And if you go to insert now in Excel, it will give you a chart like this. So just as a kind of reference, what we're looking at, in this case, each individual line is a different image from the different embryos, and left will be the apical part, and the right bit will be the basal part of the retina. And this is just intensity. And if you look at this structure now from the left to the right, from a biology point of view, if we look at the images, we know that on the top we have these 1 and 2 subregion, and then we have the cell bodies, which are very, very bright, which is what we see here. So this is the subregion 1 and 2, and then the cell bodies. And then we have a region that is slightly dimmer, and then these end feet. And this is exactly what we see here. And also what you see is that these two have a different size, and that's why the end feet are at a different position and the image ends at a different position. And if you would have selected your normalization before, they would have just been stretched to the same size. But in this case, it shows us truly that these are two different, or they're slightly different in height, these two images. I'm just going to try and stop share now. Does that so far all make sense? Anyone lost at this point? Any questions? Yes, no, maybe? It's very hard online when there's no, I'm looking at a blank screen, few faces, which is nice, but it's very hard to gauge if, people find that useful, if that makes sense. If I've lost already everyone, thumbs up, thank you, that's always good. Great, thank you. Fantastic. I think that this tool is probably also very useful to people who don't work with Glia cells. It's applicable to anything, so if you do want to test it for any other data, please do let us know. But I think that this is going to be very useful to other people as well. So if there aren't any further questions on this, I think I'll hand over to Karim, who's now going to talk with you about image segmentation. And again, yes.
Right, sorry about that. Lovely, I just share my screen. Apologies for the camera. My laptop decided it was a good time to be temperamental. Okay. Right, so we've reached the stage of image segmentation. Now, before we delve into how to carry out this step, let's talk about some of the pre-processing stages. Now, we haven't carried out all of these steps, so we talked about the subregion tool. But let's briefly talk about the others, just to make sure that we're caught up on what these things are. So firstly, we can perform deconvolution. And what that does is it ensures that we have the correct measurements in all 3 axes, X, Y, and Z. Secondly, we have the subregion tool, which we talked about, and that's to orientate the stacks in the same direction so that we can compare them. And then thirdly, so if we are working with many channels, so for instance, if we have a nuclei stone, a membrane stone, etc., then we can split the channels so that we're working with only one channel, the information that we require. Now, after completing these pre-processing steps, we can now talk about the image segmentation step itself. So the purpose of this step is to convert our original image into a black and white image, as shown here. Creating this image allows us to delineate the outline of the Müller glia, and that will help us in the next step of image analysis. Now let's talk about some of the features involved in this step. Firstly, let's take 2 of the features listed here. So we'll take contrast enhancement and median 3D. Down here, you'll see a pixelated image with darker pigmented pixels, black and dark grey, and those represent part of the Müller glia. Now towards the edge of the Müller glia, the pixels gradually become lighter, until it reaches the white background. This creates a gradient between the Müller glia itself and the background. However, along this gradient, we also reach these lighter pigmented pixels, these light grey pixels. And one of the challenges we face in determining whether these lighter coloured pixels are part of the Müller glia itself or artifacts in the background. Now, over here, we draw an arbitrary threshold to decide which pixels to accept as part of the Müller glia. So for example, over here, we have the black, the dark grey, and the grey as glia. We can use contrast enhancement here to reduce the amount of pixels within this threshold area, and that creates a more definite delineation between the black and white regions. And that makes it easier to determine the outline of the Müller glia. We can also use median 3D. So to explain this function, let's take a single pixel, for example, this one. So we'll look at the pixels around that pixel, and then we'll get the median of their values. And that then becomes the new value of that pixel. Moving on to the next step, next feature of the segmentation step, we have Z-axis bleach correction. Now, I'd like you to imagine a laser passing through layers of a tissue. As the laser passes through, it attenuates. Now, this means that the innermost layers being visualized have a lower data quality. And we can try to mitigate this by using Z-axis bleach correction, which is a function that allows us to restore data quality to those innermost layers. Another feature of our segmentation process is edge enhancement filtering. So one of the benefits of including this function in image segmentation is to smooth out the membrane of the Müller glia, particularly at the cell bodies such as over here. This function also allows us to identify finer details, such as the inner plexiform layer projections, as we can see in this image. And then finally, we have the thresholding method itself, Otsu thresholding. So there's various types of thresholding methods, but we've chosen this one in particular because it gives us the best delineation of cell bodies and projections. So now we've discussed the features included in our segmentation process. How did we assess whether these features improved the quality of the images we acquired? So here we have some techniques that we can use to validate the quality of our data. Now, there are some differences between the validation methods used in medicine and the validation methods used in biomedical sciences. So, for example, in the medical field, we can use manual segmentation. This is where an expert picks out the region of interest, either by using the slice-by-slice method or in a 3D viewer. And another method we can use in the medical field is phantom models. And this is where we create items that we know the measurements of. So, for example, in this model here, we know the inside diameter of this tube is 1 millimeter. And hence we can use that to calibrate our imaging software and accurately measure the dimensions of tissue samples. Now, moving over to the biomedical sciences field, we have a different repertoire of techniques that we can use. So firstly, we can directly compare the images just with our eyes. So for instance, here we can see in the middle that this image is different to the other two. Most likely, evidently in the fact that these Müller glia are quite incomplete. The other two images look fairly similar, but there are subtle differences. So for example, like this spot over here looks fairly more dominant than this one over here. We can also use visual assessment tools. So one of these is image subtraction. And what this does is we take one image and another image, and we subtract it from another. So then eventually we get an image which shows areas that are present in one image, but not in another. And this can help us identify which method is better at picking out finer details, such as the projections in the inner plexiform layer. And then finally, we can measure how much overlap there is between two images using tools such as the Jacquard index and the Dice coefficient. Now, both of these measurements compare the voxels in one image to the voxels in another image. And it gives us a number between 0 and 1, with 0 being no similarity at all, and 1 being totally identical. So now we've talked about assessing image quality. Let's move on to how to carry out the step itself. So I've just opened up image J over here, and you'll be glad to know this step is quite simple to do. So we'll open up the GliaMorph, which we downloaded, again into macros, and this time we're going to go to step 7. So if we drag and drop that over here, we've got the macro screen, and we'll click Run. So let's go to our I2K 2022 folder, tutorial prep. And the folder we'll use here is ZDIR. So if I just show you what's inside this folder, we go into I2K 2022, tutorial prep, ZDIR. So what we have in this folder is the Z reduced images, which if I open up, we can see is like this. So this is the image after we've done the subregion tool. So if I close this, and then, so this is the folder we're going to do it on. I would press select, but because it takes such a long time, I won't do that for the moment. Instead, what I'll do is go through what we should get after having run the macro. So ZDIR, we should get this folder called TH. So in this TH folder, we'll have this and our 3 images. So I'll just run through what they are. So MIPs, you have the maximum intensity projections of the 3 files over here. And these images we can see are the segmented images which the macro has produced. Apologies. And then once we have these images, we can then proceed on to the quantification step. And that concludes the segmentation process. Are there any questions so far? Let's have a look in the chat. It doesn't appear to be.
Karim, do you want me to take over answering the 2 questions that came through? Or?
Yeah, sure.
Yeah, okay, cool. So thanks for the questions. So the first question is from Jennifer, who said, could this also be used on branched neurons? So in general, yes, but segmentation is always the most crucial point in your data analysis if you want to perform a quantification. And so obviously our quantification that we are showing you today has been very heavily optimized for what we are looking at today. If we would even look at these data with a different antibody staining or something, we would again need to look at the segmentation and improve the segmentation for this. And similarly, if we now switch from Müller glia cells in the cerebrospinous retina to branched neurons in, I don't know, the mouse visualized with antibody stainings, this obviously needs optimization. There are some general kind of steps that you can follow through and look at in terms of segmentation, which is also something that we kind of have very briefly mentioned in our work, which is like called segmentation test, so you can have a look at that. But segmentation in itself is probably the most crucial step, and that will always need optimization, and that has just to be mentioned that you will never just be able to just apply to something else. So unfortunately, that is the most challenging thing for most data analyses. The second question is from Powell, who asks, have you tried other segmentation methods like instance segmentation? So in our case, what we do is we segment all the glia cells, as you've seen in the 3D data. And kind of the ideal world would be to get individual glia cells. But in our case, because we use transgenics, all of the glia cells are visualized and they're very, very tightly connected to each other. So we can't really say which one is ending here and starting at the next point. And so this is what we would call like identifying individual glia cells, which is hugely complex from a computational point of view. So one thing that we're trying to do in the future is to look at this from a machine learning point of view, where we use data like these, but say, we know that one cell starts here, the next one ends here, etc., and then train this for having an automatic segmentation. So this is one path that we can go through. But the second way to look at it is not from a computational point of view, but actually from a biology point of view. And that's what we've done for now. So what we did is actually inject DNA in the one cell stage in the embryos. And so what we get is a so-called sparse labeling or individual clone. So you can do that with mice, you can do this with fish, etc. So you can have this sparse labeling approach. And that's then extracting individual glia cells. So we can then not measure, in a sub-region, but actually get measurements for single glia cells. And that's what we're doing at the moment. But yeah, the future kind of ideal, beautiful thing would be to say, we know one glia cell starts here and it ends here. But at the moment, we can't really do that. And I hope that answers your question. I can't see him. I think we've lost him. Oh, no. Oh, yeah, that's fantastic. So he actually says he's trying to get a deep learning model working for instant segmentation of Müller glia cell bodies. I would love to have a chat with you about that because I looked at various different things. So if you want to catch up afterwards, that would be fantastic. Because yeah, there's definitely some overlap here. Any other questions? No? Okay, fantastic. So again, segmentation is probably the most critical step, but also the one that is the most, that's the one where you will need most adaptation to your own data, unfortunately. But it's always worth to spend a lot of time on your segmentation and knowing that your segmentation is very sensitive and robust, because otherwise all your following quantification won't really be working, or it will not be as good as you want it to be. So with this, we're going to now move on in the GliaMorph workflow. And so if we look at the structure here, so what we've done is, again, we went from raw data to have a subregion tool where we extract these subregions. We didn't have 2 channels, but 1, so we skipped this step and directly went to the segmentation tool. So now at this point, we have measured the image height, the volume and the volume percentage coverage, and these data are also in an Excel file in the TH folder that Karim has shown you. What we're going to do now is actually apply the so-called quantification tool, which will take segmented data, so binarized data, and quantify various different features. So the things that we will quantify from a biology point of view is the thickness, the skeleton length, number of junctions, number of endpoints, the average branch length, the surface, and the surface to volume ratio. For those of you who are more kind of computationally interested, the way we do is we use an EDM for this, and combine this with a skeleton. The skeleton approach that we use is the standard 3D thinning approach to derive the network, and the surface is just using the canny edge detection that's already implemented into Fiji, and this is then allowing us to extract all of these parameters. But again, for those of us who are biologists, we kind of ignore all of this and just use the quantification tool. And in this case, it's step 8 quantification. So again, you would drag and drop it and you hit run. And in this case, your input folder will be your TH folder. So in this case, you would go into your Z directory, so the one that is made smaller, and sorry, you just use this. And in here, you would have your 3 TIFF files that are then segmented. In this case, I don't have it in this one because I didn't run it. So I need to, go into this other one that I already prepared. So that's the TH folder, and the TH, so the segmentation, would be your input. If you again select, then it runs, and in this case, it will run quite a while. So I've just stopped sharing for now because it will run in the background, because especially this skeleton approach where it will do this 3D thinning actually takes quite a while to run, because it obviously takes layer by layer off to then get your individual kind of skeleton. So it will take quite a while. If you have run it, I'd say probably 20 minutes now from now on, that it runs through those 3 images, which is not a long time if you think about kind of the kind of all the computations that we do. But again, these are steps that I would personally kind of start overnight, or when I'm going to make dinner or something like that, and just let it run and then come back to it, and hopefully it has all worked. this quantification tool works with any segmented data really, so it's independent of your input data type. So again, if you would have branched neurons that are segmented, for example, in, I don't know, Arabis, Imaris, whatnot, as long as they are TIFF files, it will work on this. The most important thing is that they are all the same size, or at least more or less the same size, because obviously you want to be able to compare one image to the other, and often we can't directly do this when we are on the microscope, because obviously we have these different fields of view, different angles, differences in intensity, etc. And so this sub-region tool is probably the most crucial to actually being then able to compare different images against each other. Okay. Are there any questions at this point? No? Okay, if not, so what I would suggest is that we have a 10-minute break, we get a coffee, and I wrote down kind of a post-course reflection so we can have a little bit of a discussion afterwards. We will finish this quantification and we will look at what the data are that we get. But I think the most important thing is that you guys, you know, think about what does this mean for your work, or, you know, what does this mean for your research? So it would be great if we can start a discussion. So yeah, if you could kind of think about a few, that would be great. And if we could meet here at past, whatever your time zone is, that would be fantastic. So about 12 minutes from now, grab a coffee and come back. Okay?
Shall we pause the recording, Ellie?
Yes, please.
Okay.
I'd say we just give it a minute until people come back and then we can just restart. Okay, if people can just drop a yes or a thumbs up so we know that they are back, that would be great. Fantastic. Okay. That looks like more than half. That's sufficient. We shall proceed. So now, hopefully over the break, your quantification tool has kind of finished. Normally, it probably is still running, but we can now kind of start showing you what will the output be looking like, and we can talk through that a little bit more detail. Again, just drop any questions in the Q&A or just interrupt us. So, in this folder, the TH folder, this is what you've selected as an input. That's the segmentation output folder. So basically the binarized images, which are called TH something underscore, were now taken and we did all these quantifications on them. So that's the quantification tool. And what we get are these output folders, whereas out EDM is basically containing your Euclidean distance maps. So that was used to get the diameter or the thickness. Your out skeleton is your skeleton measurements, which is again just tracing or the center lines of your glia cells. Your out surf is the surface, so the canny edge detection that we use to quantify the surface. And out zone in this case is basically the plotting, this apical-basal plotting of the intensity using this 3D to 1D vector conversion, and that's what we call out zone. And we have a similar output here in the out skeleton, where you have sonation tool profiles already here. So these are basically these profiles that you will see here. And so at this point, I think this is where now your kind of biology knowledge comes into play. I'm just looking. So thanks, Jennifer, for the question. She just said, out of curiosity, why do you perform the 90-degree rotation tool? And so in this case, just to kind of jump between things, you could use this in between the sub-region tool or like your data and sub-region tool, because sometimes when you acquire very long data, which are not very rectangular, you just need to rotate your image first before you can do the sub-region tool. But I can link to the bioRxiv manuscript again, and there it will describe in more detail when you would use that. But in, I would say, 90% of cases, you would not need the 90-degree rotation tool, but you can use it obviously if you just want to rotate your images. Okay. So back to the actual output. So at this point now, biological interpretation comes into play, and again, your segmentation is probably the most important step, so any quantification will make sense at this point. And so what you would normally do is just really analyze the data and look at them in a little bit more detail. And so I just want to bring up some of the examples. So this is one of our data sets that we're working with. So this is fish at 3 days and at 5 days. This is the same transgenic, but we're looking at how the glia cells change over time. And so this is something that we look at as standard. Similarly, we would look, for example, at drug treatments or things like that. But one thing that we do is we plot the data, and obviously we want to quantify this, so we use statistical analysis, but also an important thing for us at this point is seeing kind of the variability. I think what we all know as biologists, like, you have some kind of spread, but obviously if some of your data would be up here and some of your data would be down there, you would know that something is not quite right. So just having this kind of intuition of like what your data more or less should look like in terms of spread is very, very important. And then obviously you can just plot your data that you have in your output. So if we just go to the TH folder in your quantification results, it will look like this. But if you just copy and paste that into a normal Excel file, it looks more like an actual kind of table. It's just as a CSV format. And what you see is that you have this output of your 3 image measurements. Again, these are your 3 images, and then you get these measurements. And then you can just copy and paste this. And in my case, I just use GraphPad, and this is what we get as visualization. Obviously, you could use other visualizations. And I think here it's also important to just have a little bit of a play with your data and see what makes sense. So in terms of, you know, how do you actually look at your data? I personally like this individual dots because then you see the spread very nicely. But also, for example, violin plots are very nice to actually show you, you know, where is like the majority of your data located and how is the spread looking like. Again, it's all a question of reference, but I personally prefer those over, for example, these boxes, because I personally don't find these boxes very intuitive in terms of like, you know, what's the spread, what's the variability, do I have any outliers, etc. And so for me personally, I would always first have a look at these individual values to see how are my data looking like. And again, from a point of biology, we would expect them to all be clustered rather closely than all over the place, at least for most of the questions. Okay, I'll just stop sharing and just have a quick look at the chat. Okay, any questions at this point, and does that all still make sense? Any biologists wanting to jump in and rectify what I said? No? Okay, right. Then we can continue with the data interpretation. And so basically at this point, we understand, okay, what happens with our data. And for this, we can now also come back to this zonation tool, where we can now start to make sense of what does it actually mean to plot these data from 3D into a 1D vector, and when does it actually make sense to look at the data like this. So at the very start, or quite at the start, when we talked about this zonation tool, we talked about normalization so that we could stretch the images to be the same size. And I just want to very briefly talk about this. So in this case, what you're looking at, again, is data at the left side at 3 days, and on the right side, it's 5 days, okay? These are the plots from the original images, so the ones that we used for the subregion tool, but they hadn't had any processing done to them. The second step is in the segmentation tool, so these are the ones that we have binarized, and they are in the folder of the TH. And lastly, we have the skeleton, which is basically your center lines of your images, which we get by this 3D thinning. And for each one of them, what we've done is we run this zonation tool once without the normalization. So we just run them on their size as they are. And then we've normalized their size to the same length, basically. And I just want to show you when this can make sense and why we kind of look at this. So when you look at 3 to 5 days, what you see is actually that these different kind of levels of intensity that show you different positions within the cells, they can change. So for example, here the cell bodies, they look like they migrate downwards as well as the end field, and that's because the retina gets larger over time. And so we see exactly this happening, especially in this first part. We also see this in this non-normalized segmentation as well as the skeleton. But if we actually normalize it, we see, okay, there's obviously still a little bit of migration, but the main thing is also that we see differences in intensity. And so while this non-normalized looks more at positional difference, the normalization allows us to look more at the intensity differences. What does that mean in terms of intensity? And one thing, for example, here is also that obviously it's important that you kind of compare your values against each other, because if you just look at one plot, I always find it hard to interpret what does that mean. And so when you just look at 3 versus 5 days, you can already say, okay, there are actually differences happening. And this is when biology makes sense, when you compare 2 states against each other. And so this is just one example of when we would use non-normalized versus normalized. But again, I would run both of them and then see what makes sense for your data. And then in terms of kind of how we visualize the data, again, it's various different ways. But I've started to actually use depth coding for my 3D stacks because I find it very intuitive to say these are 3D data. And again, it's just a question of normal or like visualization, but I think that this is something useful to show people that we work with 3D data, because often you could assume that you work with 2D data, but you actually don't. And then basically all that I've done down here is just plot all the data that we get out of the GliaMorph toolkit and just look at them against each other. And so basically when you run the tool, just rerun it now, you will have all of these data at this point and can look at them. Okay. So, any questions? Any ideas from anyone? Any input?
Hey, Elisabeth, you're gonna get tired of seeing my sideways picture. I'm not sure what's going on with my camera today. I'm really excited about this. I mean, this is really cool. And certainly I'm going to play with this because for me, in my analyses, I see several different ways that I could use this. So your Müller Glia, are very vertical. But the, excuse me, the Glia that I'm going to be looking at, like astrocytes, have distinct morphology that when they're resting, they're in one shape, and when they're actually activated, they're in a different shape. So have you guys had any thought about doing orientation of the branching points?
We haven't looked at orientation of branching points, but there are 2 things. So A, we have started to look at actual orientation within the cells, so in terms of alignment and order, which is something where we actually collaborated with people from KCL, and I'm just trying to find that section in the bioarchive.
I'm definitely going to be reading that, so no worries.
I think it's just generally interesting because it's a very different approach as well, so I just want to very briefly show that anyways, because it's a good thing to bring up. Just share the screen again. So again, in the bioarchive manuscript, we have looked at orientation so it's not orientation of branching points rather than overall orientation. And in this case, we use a tool that is based on the Fourier transformation, which is from Stefania Marcotti from KCL. And this works on 2D data, but basically we kind of wanted to know, kind of what's the alignment, because we know within this IPL we have our protrusions more being aligned sideways rather than the overall morphology being upwards. And so we kind of want to know what is happening over time, and we see that over time, especially in the IPL, what you see is that you get more of this sideways alignment, so you get more of this horizontal procedure, so the protrusions start to be more aligned to the side. So that's one thing that we found. A, it's very quick to use, and it does need only little adjustments, so you could look at that. But also B, another thing is that you can actually look at its shoal analysis, where you basically plot your center points, and then you can measure how many branching points do you have the further you go outwards, which is again something in Fiji that you can do in 2D and 3D. I personally find it mostly sufficient in 2D, to be honest, and it works very fast in 2D as well. Whereas 3D.
Favorite tool for that on Fiji?
I think it's literally called Fiji. Let me just wait. It's somewhere. either it's in the neuroanatomy toolkit
Okay.
or I just can't move around at the moment on this one.
So I used to use a legacy tool, but I'm trying to get away from some of the legacy tools.
The problem is there's so much in Fiji that you can do. It's so easy to just lose.
Yeah.
I think so. So yeah, if you go to the neuroanatomy shoulder analysis from image, so that's what I've used before, and I found that to work quite nicely, because again, you need to place a manual center point, but yes, I mean, you know, you kind of want to go away from manual things as much as possible, but I think especially with astrocytes, right, you could determine, okay, where is my cell body center, and that's always my center point, or things like that. So it's nice to find the standardization, I think.
So I was asking earlier about branched neurons because one of my other analyses was with Purkinje neurons.
Mm-hmm.
And they're large and they're heavily branched, and the segmentation of those, I was having a lot of difficulty actually achieving a good segmentation with those. And so trying to find a good segmentation that can be batch processed. Yeah, like I said, I'm very new to this. So learning on the go and on the fly as we speak, as we say.
I'm happy. I'm happy to have a look if you want to, you know, catch up, or like, you know, send me some data, something. I'm more than happy to, you know, have a look and kind of, you know, look a little bit more in detail in that, because I think the most important thing is to get a pre-processing that makes your images really so good enough that the segmentation itself is not biased in a different direction. Because especially with neuron data and, like, you know, data like these, the problem is also that you normally have less data than background, and that is obviously also a challenge, right? And if you then have, for example, antibody stainings where you have a lot of background in your background, that's just a huge challenge. And I think that's just so you need a lot of pre-processing to get rid of your speckles, your fluorescence, your background, and then once you have that, the actual segmentation is not easy, but at least it's like so much more easily applied to a batch, because at the start all your data will be so different, and that's obviously a huge challenge. So yeah.
Okay, thank you so much.
We've all been there, and I think we're all always there with new data as well. So yeah, absolutely understand that. Great question, thank you. Do we have any other questions or comments? Anyone thinking about applying into their own data, having any questions right now? You can pick our brains as much as you want.
Elisabeth, I do have a question. Hey, so when you do the volume analysis, do you get it for each single Müller glia, or is that for the entire region of interest that you have taken out?
So in this case, it's for the entire region of interest. But again, so you can either do your clones. So that's one thing, how you get individual cell volumes, or another way is also that you can kind of approximate the number of cells and then just divide it. So for example, the region of interest that we look at is on a standard about 12 cells, and if you divide it more or less by that, you get approximately the same number. But again, it's not 100%, because obviously you cut off cells as well sometimes, so it's more like an overall volume / density.
Okay, great, thanks.
Thanks. Okay, last chance if anyone has questions. No? Okay, so if there are no more questions, I hope that this was useful to you. I'm happy to kind of stop now, and if people have questions, we'll be around in Gathertown. But yeah, it was a pleasure to having all of you here. I hope it was useful. Again, if someone of you want to send me some data to have a look at them, we're more than happy to do that. I want to thank all of you for joining, and a huge thanks to Sara, Karim and Eva-Maria for being absolute amazing co-hosts. And again, thanks for all the questions. If you have anything coming up later or any comments, please feel free to just get in touch with us. So thanks everyone. Have a lovely afternoon. Bye.